Recently, scientists placed human brain cells in a lab dish, connected them to a computer, and taught them to play a simulated video game. In a separate experiment, researchers monitored lab-grown neural tissue, successfully using the electrical activity from those cells to control the movements of a robotic spider. This field is called Organoid Intelligence, or OI. It is the merging of living human brain cells with hardware and software systems. You might hear these called mini-brains in the press, but right now that is an exaggeration. These are microscopic 3D cell cultures. They lack blood vessels, whole brain structural organization, and consciousness. But this creates a very specific trap. The traits that make these cell clusters useful for curing diseases or building biological computers are the exact same traits that create moral hazards. If scientists want to understand how Alzheimer's destroys memory, or if they want to build a biocomputer that outpaces silicon, they have to engineer these organoids to live longer, be more connected, and capable of causal learning. This graph maps the core tension. As the organoid's scientific utility increases, the ethical risk rises alongside it. Eventually, these parallel lines cross into a shadowed, undefined zone because we do not know exactly where the threshold of consciousness sits. If we wait for a lab experiment to show us where that line is, we will have already crossed it. Society has to decide what the rules are before the science finishes developing. Right now, labs are plugging these organoids into microelectrode arrays. They stimulate the cells, monitor the electrical readouts, and train the tissues to respond to feedback. Simply being grown from human DNA does not mean a cluster of cells has moral standing. A skin cell in a petri dish has human DNA, but we don't grant it protection. Moral status, the idea that an entity matters for its own sake, is tied to observable capacities. The relevant questions are, does it feel pain? Does it have agency? Does it have a subjective experience? Without a definitive scientific test for consciousness, researchers cannot point to the exact moment a biological tool becomes a biological subject. We are moving toward a boundary in the dark. To reach those advanced capabilities, scientists have to solve a biological limit. Lab-grown organoids don't have blood vessels, which causes them to starve and die before they can fully mature. To get around this, researchers are transplanting human brain organoids directly into the brains of living rodents. The animal's body provides the vascular system and nutrients the human tissue needs to survive and grow. Putting human neural networks into an animal's brain creates a new set of problems. We have to monitor whether that human tissue alters the host animal's natural cognition, its behavior, or its capacity to suffer. By building human-animal chimeras, we blur the lines of species identity. If an animal gains enhanced problem-solving skills from a human organoid, we have to determine if that enhanced animal now commands a higher moral status. This ethical web also reaches back to the source. Every single human cell line used to grow an organoid started as a biological donation from a living person. Standard medical consent forms are designed for one-time procedures. They are insufficient for dealing with unpredictable biological technologies that change over time. If a patient signs away a tissue sample to help study a disease today, they are not necessarily agreeing to have those cells power a corporate biocomputer or drive a robotic system 10 years from now. Because these organoids carry the donor's genetic information, the donor's right to privacy doesn't vanish when the cells leave the hospital. The biological material remains entangled with personal identity long after the donation. The sheer number of moral hazards here might sound like a reason to stop the research. But walking away means walking away from tools that could finally model brain injuries, test dementia treatments, and lower the energy cost of computing. Instead of a ban, researchers are proposing a framework called cautious enablement. It allows the science to proceed, but only under a system of anticipatory governance. The first pillar of this governance is the use of embedded ethicists. Instead of calling a philosopher after an experiment makes headlines, labs would integrate ethics experts directly into their teams to evaluate an experiment's moral weight. The second pillar is dynamic consent. Donors would maintain an ongoing relationship with their biological material. If a future experiment crosses their personal boundaries, they can simply hit the brakes and completely withdraw their tissue. The third is graduated oversight. This means regulatory rules would tighten based on what the organoid can actually do. As the tissue gains complex capabilities, like causal learning, the legal protections scale up to match. Technology does not just happen to us, we build it. With organoid intelligence, we can see the hazards from a distance. We have the opportunity to build the ethical guardrails right now, before the lab dish wakes up.